David, how long does one treatment with the virus packaged RNA last in the body?
So the one we published was a 4-month treatment. So we did the injection and we look at the efficacy, stability, safety, blah, 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 during the time of 4 months. The virus is much more stable than these. There are reports in primates showing the persistence of AAV for more than 8 years after a single delivery. So it's a very stable virus if the tissue is not a tissue that replenishes itself. So for example, the skin, where we have new skin cells very frequently, there will be lost very soon. But in a tissue like brain or muscle that normally is a turnover rate very slow, the virus is kept for a very long time.
Have any other researchers successfully reproduced the results that you obtained with the virus as yet?
Which result? The therapeutic, let's say, result that I describe you, there is another group in the US that have used also the FRG1 mouse, and they obtained similar result to what we obtained at Scott Harper group from Ohio State University. They didn't do a systemic delivery like we did. They did a more simple approach. They did just intramuscular injection, so they were unable to look at the long-term safety-related issue that we looked, but basically they obtained exactly very similar result to what we obtained. Yeah.
So if I've sort of understood this correctly, you're sort of saying that somebody like Bill Moss here, in 10 years, he might not need somebody to help him walk about.
That's the hope, of course. Let's say that since 5 years from now, we are in a much better position because we have reasonably good candidate for the disease, and we have a group developing several different possible therapeutic approaches for the disease. So we are no longer simply looking at the possible cause. We are already developing and testing possible therapeutic approaches. So there's people like us that have used viruses. There's people like Michael Kaiba that is conducting, that I think is also supported or has been supported by the foundation, which is using a small molecule inhibitor screening, like has been described before. That's actually the question you ask, to look for molecules directly affecting the activity of DAX4 to prevent DAX4 activity. There's people that is using drugs to treat not the initial cause of the disease, but possible downstream effect. There's trial going on in France for antioxidant for FSH. So there is people actively involved in testing possible therapeutic approaches, which is much better than what was 5 years ago. The hope is that, yes, in 10 years we will be there, but...
This particular virus that you're using in minus, can that virus reproduce in humans, or would you have to find another virus?
The virus is actually naturally present in our body. So it's already there. The virus is, as I said, part of the reason why people likes it is because it's a very safe virus, because everybody, or most of us, have encountered this virus during his life without having any particular problem. So the virus is capable, the natural host of the virus are human beings. So the virus can go inside very efficiently inside human cells and infect human cells. And there's quite a few clinical trials already going on testing the efficacy of this virus, similar viruses, for a number of genetic diseases, including muscular dystrophy. So there are several clinical trials going on at this time for Duchenne muscular dystrophy and Limb-Girdle muscular dystrophy, testing the safety and efficacy of this virus for therapeutic purposes.
That's tremendous.
Yeah.
Thanks very much, David. Just wondering,
So I think they are both important. I think they are both important for the disease, and actually we have result in the lab connecting them, putting them in the same pathways, which I'm not ready to disclose, but which, as I said, so suggest that they actually collaborate in driving the disease. Yeah.
Davide, is this the first time that researchers have shown that the loss of DNA or genetic material can cause an increase in protein production associated with the disease?
For a genetic disease, yes. So we were, again, this is going back when I was a postdoc. The initial discovery was published already 10 years ago. We were when we published that paper, that was the first demonstration of a genetic disease caused by the loss of a non-coding piece of a repetitive sequence of DNA. Yes.
David, you said there was some since then.
I didn't hear you. Sorry.
Are there others now that have been shown?
Yeah. So there are other diseases in which it has been shown. This is a more general discussion about copy number variations in our genome and susceptibility to disease. So you may or may not be aware of the fact that all of us differ for the fact that we have part of our genetic material, DNA, that may be present in different copies in different individuals, much similar to the disease locus, FSH. So as you know, the disease locus, there is a repetitive sequence which is called D4Z4, which is present in different copy number in the different, even in the general population. So in healthy subjects, you can find between 11 and more than 100 copies of this repeat, while in the patients, you know that you have less than 11 units. This is not the only example of this particular sequence. In our genome, we have several other regions that are characterized for the fact that they are present in different copy numbers in different individuals. And the variation in the copy number of some of those is not associated with a disease. There are instead several other diseases in which variation in copy number has been associated to disease. There are genes that are involved, for example, with the ability of HIV to infect the host that are undergoing copy number variation. And so different copy numbers correlate with the susceptibility to infection from HIV, for example. Form of cancers in which copy number variation correlate with the progression of cancer in this disease. So there's quite an increasing amount of examples of that sort.
Okay, thanks.
Thank you.